Alright, so, yes, welcome. Today we're doing the transformation, and transformation basically is changing the genetic makeup of our E. coli by having the E. coli take in the plasmids that we made yesterday. Now, how do they do that? So, we have them sitting on ice right now. I pipetted the plasmids into them, did that little tapping mix, and what's going to happen is, you know what, I'm going to grab our little mascots here. E. coli sitting, so here's our Eli E.coli sitting in the ice right now. What's going to happen is the plasmid DNA is basically going to sit right on the membrane of our Eli E.coli. That's what this incubation on ice is for 30 minutes. And because of that, we're just gonna, it gives it time for it just to sit there, get ready. It's basically priming up these cells, because then we're gonna heat shock them. I know that sounds terrible. We're heat shocking living things. But what happens when you heat shock it is we're going to put them at a higher temperature than what they're used to. So E. coli usually grows at 37 degrees Celsius. So once again, science, we use metric. Fahrenheit 37 I think is around, oh gosh, like body temperature. I want to say it's around 97 degrees Fahrenheit. I'm wrong. I'm sure it's close. It's close to that. It's like a nice little warm sauna. And we're going to put them actually at 42. So that is higher than what they're used to. And what happens during this time is little pores end up coming into the membrane of the E. coli. So it's basically like starting to get very upset at this temperature, but we only do it for a minute. So it's not sitting there for a long time, because if you sit it for too long, the poor E. coli is going to die. Just because it doesn't like that temperature, those pores are going to keep happening in the membrane and everything's just going to spill out of the cell. So the amount of time that we put it at 42 degrees Celsius isn't enough to kill all of them. Some of them might not survive, but a majority of them are going to be okay. And so once the pores happen into the membrane, any DNA that's sitting on Eli E. coli, so this DNA that we're basically sticking to the membrane right now, so it's sitting like this. When a pore comes in, the DNA is going to be able to go into the E. coli cell and then be incorporated into our bacteria. And that's basically what the heat shock does. So after a minute at 42 degrees, we stick them back on ice just to stop that heating, just quickly stop it so nothing else happens to them. And that's only for 2 minutes. And then we basically pamper them. We basically say, sorry for doing such terrible things to you. Here you go. We're going to give you the food that you love. We're going to grow on a nice little plate. We're going to put you at the temperature that you love to grow at. And that will make them happy. And if they manage to take up the plasmid that we made, they'll grow on the plates that I'm going to put them on. I will show you the plates later, which are warming up in our incubator right now at 37. So we have a standing incubator just for bacteria in the lab, always set at 37, because we do a lot of plasmid cloning. We've done so much plasmid work in the lab since I've been in here. So it's always on, always ready to go, and it's just for bacteria. Excuse me. I have a little frog in my throat. So, yep, and then it will grow at 37 basically overnight, and then we take it out, put it in the fridge, because I won't be able to work on it over the weekend. I will be here on Monday, and hopefully be able to show you, like, quickly what we do after it grows up. So, any colonies, hopefully we'll get colonies, and basically they should look like little white, dots. They're like good-sized dots, like, pretty good-sized dots on the plate. And those that grow on the plate means that they've taken up the plasmid, because the plates that we have an antibiotic resistance on them. Or the plasmid has an antibiotic resistance on it. The plate has an antibiotic called ampicillin. Excuse me, I need to cough. Just a second, I don't want to cough for you guys. There. Hopefully that's better. Sorry, just a little frog in my throat today. So yeah, so basically that's what we're doing today. We'll let it sit in a shaking incubator. So Basically, it's an incubator that has a little rotator thing on the bottom of it that's set at 37 for an hour, and that's when we plate it and stick it in the incubator for overnight in that case. But yeah, so sorry, getting right into the science this morning, but hopefully everyone is okay in chat. If you're having a good day so far, that would be awesome. Like I said, I'm starting early this morning. If you have any questions, please feel free to put them in the chat. I'm always open for questions. If you want to start chatting about something else, we can start chatting about something else. Just because we have time to kill today. Lots of time to kill in this procedure. So I guess I'm looking in the chat right now, see if there's any scientists going. Yeah, there's a couple. Got a couple of scientists in the chat today. But if you don't want to talk science, we don't have to talk science in that case. Because a little bit more about myself. If you missed it yesterday, I work in the lab. I'm a lab manager of a wet lab. This is my bench. And this is where the magic happens. But outside of that, I usually play video games. It's my way to de-stress, and most of the time I play simulation games. So, Sims, I love 2 Point Hospital, Project Hospital, what's another one that I've been playing recently? Let's Build a Zoo. So, indie games. I usually play indie simulation games outside of Sims and the 2 Point series. I've been playing a little Fall Guys, but I'm terrible at it. Not the best. I also have 2 kids, 2 cats. So I'm a baby mama and an animal mama. Big anime manga nerd since a long time ago. I also cross stitch as a hobby. It's good to have hobbies, obviously, outside of work, since this is work, but I still love what I do. And I'm glad that I can share it with all of you right now. And what else? Let's see. What else am I doing? Oh, we always watch so much kids TV because I have little kids. So I've seen so many shows. So many kids shows. But yeah, so if you guys would like to chat about something, just lurking, I don't mind lurkers. I love lurkers. You don't have to be forced to chat. I'll never force you to chat because I am a serial lurker in channels when I'm watching Twitch. Just because I usually am there just for the vibes, just to enjoy what they're doing. Usually watch gamers on there, like-minded gamers, mostly The Sims, because I enjoy the stories and it's usually fun and comedic just because the game can be that way. And yeah, so don't have to chat. You don't have to chat. I can just sit here and talk if you just want to hear my voice. But yeah, so as far as science goes, I just explained it, but if you have any questions, we can talk about that. As far as the procedure, or what is E. coli, or all these jargony terms that I seem to throw out there every once in a while, I'm here to help you understand what I do as a scientist, what molecular biology I'm usually doing. So please, don't be scared to ask questions in the chat. And if I don't know something, I will look it up. No problems there. I can admit that. No problems admitting that. I don't know everything. And I feel it's impossible to be able to know everything in this field because it's always changing and definitions change. Even the long-term definitions can change because biology always has exceptions to rules. If you can say that. So yeah, so we've got about 19 more minutes left. Oh, the timer actually shows up pretty well on here. Yeah, so science. I'm gonna say, I guess the biggest news in science last week was Gregor Mendel's 200th birthday. If you guys are not familiar with Gregor Mendel, he is basically the father of genetics. If you learn genetics in undergrad, I'm sure you've heard of Mendelian genetics. Oh yes, sure, Dizzy, not a problem. We are transforming the 2 plasmids that we ligated yesterday. So that's what we're transforming Eli today. So it's just the 2, the A and the B and the C and the D. So I'm just continuing with the cloning procedure that happens in the lab. So, good question. So hopefully it'll look good. It'll look good. I'm excited to see what the plates look like and to share what the plates look like with you guys. I will be sad if there's nothing on it, but it happens. It happens. Then we just do some troubleshooting. Yay, science and troubleshooting. Lots of problem solving. It's a big skill when you're in the lab, because not everything will go right. And even outside of wet lab, I know that bioinformatics, same kind of thing, you're troubleshooting your programs constantly. But yeah, so what was I saying? Gregor Mendel. Yeah, so if you've heard of Mendelian genetics, basically it's the basic inheritance that you usually hear about. If you remember Punnett squares, you know, the square. Oh, ooh. You know what I can do? I've got my fancy notebook right here. And maybe this will jog your memory. Here's my fancy smancy notebook. Alright, I'm gonna make some my fancy smancy. There. Hopefully you guys will be able to see it. I messed with the focus so that you could see up here where I'm usually working. So it might be out of focus here. But if you guys what a Punnett square looks like. Basically this kind of square dealy. Right? Like that. And then you usually have these letters that show up. Does that ring a bell? Punnett squares, for those who have seen them before, and then you usually fill it out. Basic genetics test when you're doing Mendelian genetics. And this is a classic case of heterozygosity, so meaning that two different alleles, and this is an allele, this is an allele, and could show up as a phenotype in this case. So there's a Punnett square. So when I'm talking Mendelian genetics, he's the guy that came up with this loveliness right here. And he worked with peas. And he was looking at color and size. So short and tall. And he was able to figure this out 200 years ago. Crazy, right? Crazy. So yeah, he just turned 200 years old. I can't believe it. It's amazing. But we didn't figure out about DNA, knowing that DNA was actually hereditary until oh gosh. Oh no. Alright, scientists in the chat. When was DNA? 50s? Excuse me, I have a genetics PhD, but that doesn't mean I remember everything. Shh, don't let anybody know. But yeah, so we didn't learn about DNA for a long time after this was figured out. But yeah, so my fancy notebook. Wanted to pull that up. So that was like the big science news as far as I know last week when it came to genetics. I'm gonna put my notebook back in case you want me to. Or I'm moving my here's my gloves, my glove box. There you go, off to the side. So if you see something right there, that's just my glove box that's hanging off to the side. And I can pull this back down here. There we go. But yeah, so let's see. New chatters in the room. Yes, to anyone that's come in that's new to viewing my stream, welcome. I am NeoQStar, aka Dr. Jess, however you would like to call me in chat, scientist at the bench. And today we're doing transformation of plasmids that we ligated yesterday. So we put two pieces of DNA together in a circle. See, I'm doing it slowly. Not this. And we're putting them into transforming them into E. coli. So we're basically cloning in a molecular biology technique. So yes. If anybody knows of any other science that happened and would like to chat about it, that's fine. I'm just gonna keep on talking because we've got a little less than 15 minutes until I will do the next part of the procedure, which is the heat shock that I explained earlier, where we're going to open up the pores in the bacterial membrane, get that DNA that's sitting on the membrane right now into the cell, and then we're gonna make them all happy afterwards. So they're not gonna die on me. We don't want them to die on me. We want this to work. We want them to grow on the plates so that we can have the plasmids that we're looking for. And it's a procedure. I mean, let's see. So the next thing after they would plate, I'm just going to tell you how it usually goes. After you plate them, after they grow overnight at 37 degrees Celsius, you will pick colonies, which should be dots on the plates, and then you grow those up separately, because each colony is going to be a separate bunch of E. coli that has taken up hopefully just one plasmid. It is rare, but they can take up more than one copy of a plasmid, so more than one plasmid can get into there. They should be the same plasmid though. We're not mixing plasmids here. That's why we have two separate tubes for each plasmid that we did. And after that, you basically extract the DNA from the E. coli that you grow up, so the colonies that you grow up, and from that DNA you check to see if what you got is correct. And that involves using enzymes called restriction enzymes, and those cut DNA at certain spots in the actual sequence, in the actual base pair sequence. And I will explain that more when we actually go into it. But yeah, so like I said, we're doing heat shock today. There's actually a different form of transformation that you can do. It's called electroporation. And when you do it that way, you actually use a machine that sends an electric current into the cell. So that's still harsh on the cells, and it will open up the pores in the membrane using electric current, but it's very fast, so it won't hopefully kill them. Although sometimes I've done electroporation before, and the machine makes a noise when it actually sends the current through, and it has a very distinct noise if it all of a sudden was too high and it kills all of your bacteria. It's a very harsh noise, and that's when you're like, uh-oh, something happened, and all my E. coli got zapped. They're done. So that's always a sad sound, and then you gotta figure out why that happened. So with heat shock, we don't have to worry about it. As long as you pay attention to the timing, you keep it to a minute, we're good. Don't leave it on there for 2 minutes. Nope, you're gonna start destroying your poor bacteria, and that's not what we want. So that's why heat shock is good. Plus, it doesn't require an expensive piece of equipment in order to do it. So yeah, if you've ever hear somebody talking about electroporation or heat shock, those are just two different ways of transforming bacteria. E. coli, and for us, we just use E. coli when we're doing our stuff. But we actually make the E. coli here in lab. It's just a simple process of we're constantly propagating the E. coli ourselves, propagating meaning continually growing them in our lab, and it's really simple to do. So if anybody ever wanted to try to do it in their own lab, procedure is out there and very easy. All you need is a centrifuge. I'm sure you guys have heard of a centrifuge before, hopefully. They're the things that spin extremely fast, extremely fast. That can get down to 4 degrees Celsius, so refrigerator coldness, and have the right chemicals. So these guys are in a specific buffer that helps them basically survive going through the heat shock. So if you hear of chemically competent cells, that's what that means. That means that they can go through heat shock. And that's because the chemicals in the buffer help them survive the heat shock process. If you wanted to do electroporation, they have electrocompetent cells. The buffer makeup is different, as you can imagine, to help them survive the electricity pulses that are in there. So if you ever hear those terms thrown around, that's what they mean. So we make our own chemically competent cells. And you can also buy them, obviously. They have them from different companies. It's just easier for us to do it. It's pretty inexpensive. All you need are the components of the chemicals, a centrifuge. You're good. You're all set to go. And plus we don't have an electroporator. So we make chemically competent cells. So that's basically explaining what these guys are. What else? There was something else I wanted to talk about. And it slipped to my mind as I was talking about chemically competent cells. How we make them in the lab. Oh yeah, moving forward with it, so we basically make all the stuff that we use in our lab. And the plates that I'm going to show you, what we use to what we're going to put these guys on, we make those ourselves as well. And it's just something that you get used to when you're in lab. It's easier to sometimes make it, especially when it comes to plates. It's a little bit of time, but that's okay. That's alright. It saves you on money, and it's mostly just waiting around. So that's another thing that us scientists like to do. Not like to do, but we kind of have to do. Wait. Other than moving colorless liquids from one place to another, we also wait. We do incubations. This is what this is. We are incubating at this time, basically waiting for something for a specific amount of time to move forward with the procedure. So we do a lot of this as well. And another thing, waiting for things to thaw. So when things are frozen, wait for that stuff to thaw, and waiting for centrifuges to finish thinning. I feel like that sums it up. That basically sums up what most of our time is done outside of doing experiments. But yeah, that's what we do. That's what we do a lot of the time at the bench, is just that and colorless liquids. But today we get to see a non-colorless liquid, which is awesome. And it's the media that we use. So it's this media right here, called SOC media. I don't remember what it stands for. I can look it up if you're really interested. But that's the media that we're going to give our cells after they've gone through the whole heat shock process. And that's the one that makes them feel better, gives them the food they love, gives them that nice cozy kind of feeling, like, I guess, like a really nice house, essentially. Like, we put you through the terrible heat shock, we're gonna pamper you now. And this is the media that you pamper them with. Make them all happy again. And then we're gonna throw them on an antibiotic plate. And those that have the plasmid will live. Those that don't have the plasmid will not live. So any of the colonies that we see on the plates are the ones that have the plasmid in some form. Now, what can happen during this process is sometimes with this process with the plasmid, you can have some rearrangements happening at this point, and this is due to sequences within the actual DNA that can be troublesome. And by troublesome, I mean they can be repetitive, meaning a lot of the same base is there. So when I'm talking about bases, I'm talking about DNA bases. A, T, C, G, those are the 4 bases that happen in DNA. And if you get repetitive of, like, say, a bunch of A's, a bunch of T's, a bunch of AT's, GC's, you know, same kind of sequence in a row, it can cause the DNA to get a little unstable. And this can happen with very difficult vectors. Remember, a vector is basically the backbone that we use during the plasmid ligation that has the antibiotic resistance in that case, and everything in order for it to grow, hopefully. And the insert is the gene that we were putting in there, so we're putting a new gene into it. So yeah, sometimes the backbone can have these sequences and you can get all kinds of funky stuff happening. If you're using vectors that could be used in viral production, which people use it, and that's a way to transform human cells or other cells, those can be an issue just because they have a lot of repetitive sequence. So you can see some funky stuff happening after a transformation if you have these kinds of repetitive sequences within your plasmid. So that's why we check them after we get the DNA, and checking them with restriction enzymes so you can have specific patterns that show up when I run a gel. I know these are all jargony terms, but I promise you, we're gonna go through it together. And a gel is, trying to think, like this little square thing, and you run DNA on it. It's pretty cool. Very fundamental thing in molecular biology is running gels to check on DNA. You can run gels to check on RNA, which is, I'm sure you guys have heard a lot about RNA with the COVID vaccines coming up that are mRNA based. And you can also run gels for protein as well. So kind of like the big three that people usually work with on a wet lab bench. But we do DNA. we run DNA on gels, and we'll go through that process. I'll show you how it works, and hopefully we'll get what we want. With these, unfortunately, I have to tell you, we've had some funky things show up, and so this is a cloning or recloning process of these, so I can hopefully get one that looks good, that we're supposed to have, that has the correct pattern when I run it out on a gel. So if it's not there, it's alright. We'll try something else and move forward. And then you get to see, nope, science isn't 100%. Weird things happen that we can't control. I can't control how the plasmid's gonna go in these cells right now. It's all up to the E. coli at this point. But yeah, we just try our best. Try our best. And then we can go from there when that happens. But hopefully, fingers crossed, we'll get what we're looking for. And we got less than 3 minutes now until I will do the heat shock. I unfortunately won't be able to show you the heat shock, just because, once again, this camera right here is fixed, so it can't move with me. But I'll just let you guys hang out with the music at that point. It'll only be about a minute, so I won't leave you for very long, and then I'll be back, and we can move forward with the procedure, and then we get a whole hour for science talk. Anything you want to talk about in chat, I welcome it. I welcome it. Questions, of course. Please do. Please do. Lurkers, like I said, I love lurkers. I'm a serial lurker in many channels when I watch Twitch, so I don't mind it. It's a good vibe, as long as you don't mind listening to me talking. It's all good. But yes, if you would like to share something in chat, then go right ahead. But I hope everyone's having a good day so far. It's Friday for me today, so yay, the weekend's gonna be here. And happy Friday to those of you who it's Friday, if it's already Saturday for you. I think. I'm not sure. I always try to remember what time Australia's on. If there's any Australians watching this, I think it might already be Saturday. I don't know. I apologize if I'm very off on this. It's alright. It's alright. But yes, happy weekend. How about that? Happy weekend. Alright, I'm going to grab my lab coat, put it back on, put more PPE back on. And then we can get ready to do this heat shock. Probably hear me putting my lab coat on. I'm not sure though. Like I said, I was messing with the gates today, so hopefully my microphone hasn't been going out too much for you guys. Let me know if you do hear that happen and I can change the gate to be less strict. I just didn't want heavy breathing showing up on my stream again. As you know, as much fun as it is listening back to myself, which is not, not at all. I do not like listening to myself talk on a recording, but I did notice that yesterday, and then realized that I hadn't put the gates on, the noise gates, on my microphone, and that would be why. So, alright, 30 seconds, and then we're gonna go heat shock these guys, get that plasmid DNA into the cell for a minute at 42 degrees Celsius. Then I'll be back to the lab, and we'll continue with the procedure where we just let these guys sit for about 2 minutes, and I will add the pamper media. I'm just gonna call it pamper media. Put it right here. Alright, let me get this set up right now, and then it'll be an hour at 37. Alright, I'm gonna leave you guys very briefly and go do this. Alright, I have returned. So, what I just did was a heat shock, and now they're sitting on ice for 2 minutes to stop that heat shock, stop those pores from being made in the cell membrane right now. There we go. Now, so we're going to be using the P1000, the one that goes up to 1,000 microliters today. So bigger volumes, much bigger volumes than yesterday. I'm going to set it to 250. You guys can see that. I know it's a little hard with light sometimes passing through. Get these out. And then my amazing. So we let them shake in that shaking incubator that I talked to you about. So we don't have a rack in there that's for these guys, but tape works great. Tape is awesome. We love tape. Like lab tape, it does many things. It's kind of like the duct tape that you can show up in lab. It does awesome things. So we just tape it to the bottom of our rotating or shaking incubator. Works fine. And we shake them just because we want to get them moving within our lovely pamper media. They don't like to sit in a tiny little ball, so eventually all the E. coli will settle in the tube, and they don't like that. They like to be mixed up. They like to be away from each other when they're in a tube like this. So we just let them shake so that they get happy and enough of the pamper media moving through. And then, for an hour. So we'll shake it for an hour, and then we'll put it on the plates, the antibiotic-resistant plates. So there is no antibiotic in this. Like I said, we want to pamper them. An antibiotic, if I added that, it would not be a pamper. It would not be happy at all. Alright, so that was 2 minutes on ice. So now we can take them off. Put them in the plate. So you can kind of see it. You can't really see it, it's just cloudy. It's cloudy, it's not transparent like water when you have the cells in there, but they don't have a different color. But we're going to add 250 microliters of our pamper media to these guys. And I'm holding the media like this just so nothing floating around in the air will contaminate this, because there's no antibiotic within this. If a bacteria floating around in the air of the lab gets in here, it'll contaminate this whole thing. It'll turn nasty gross. Nasty gross. And I want to be able to use this multiple times. So that's why I was holding it at angle. It makes it harder for things to just fall in. But there you go. So that's what it looks like when we add the media. Now, my amazing lab hack. I'm gonna tape these guys together, make sure that they are closed. If they are not closed tight and we put them in the shaking incubator, there goes all our bacteria all over the incubator. No fun. It has happened to me before, and then you have to clean it up. And a shaking incubator is not fun to clean up. So we're gonna go bring these over to the shaking incubator. Hopefully you guys can still hear me. Sometimes it'll cut out. We're gonna put them in the shaking incubator. Start up the shaker. There we go. And so they'll sit there for an hour. So we've got an hour to spend with each other. I hope that is okay. But now I can get rid of my ice. I'm all set with the ice, so I'm going to go put this in the sink and get our ice bucket dry for use later. But we're all set with this. I'm going to take off my gloves, take off my lab coat. Oh. And I'm going to wash my hands because I always feel like I handle more bacteria this way. Peace of mind, peace of mind. Get the good hygiene going on. And then yeah, so we'll let that sit for an hour. And I always make sure I start my timer, because I've definitely done that before. I'm like, oh, I totally started my timer, and I didn't. And then I'm like, oh no, oh no, how long has it been? educated guess, I guess, at that point. It's never fun when that happens. Never fun when that happens. Oh, too many times. Too many times has that happened. So, always good to double check that you started your timer, in this case. Alright. I might just bring back our mascots so you guys can see mascots for a little. They're hanging out. There we go. Eli E.coli and Bert and Ernie are double-stranded DNA. Working with both of them today. Isn't that great? We get to actually see our mascots in action, essentially. But yeah, so, okay. So for an hour, alright. Ooh, do we want to talk some more science? Do we want to talk about something else? How's chat feeling today? Just want to hear me talk about something else? What do you think? Yes. I might just start chatting about gaming. Just because I'm excited for the weekend. I can play some games. In this case, just to show, even though I'm a scientist and I work at the bench and we can talk science, I don't talk science all the time. I know this is only our second stream, but sometimes it's hard to kill an hour with just science talk. I can tell you that. I can tell you that. But if someone was interested in learning about something else that I've been talking about, or something outside of this procedure that you've heard before and are just curious about it, that's fine too. I'm all for it. I'm all for it. Or you have questions about other things we might be using in the lab. Like I showed off the pipettes yesterday, which we just used this one, right? And then the racks. Of course, I brought in Ice Bucket and talked about centrifuges today. But I can always talk about that in this case. But if not, I might just out about some games that I like to play and see if anybody's heard of them before. I would love to talk about more if you have. I'm always looking for some other more gaming friends as well in here. And also, oh, I want to plug this. So I've become part of the Gamers community, and I post when I go live on there as well as on top of Twitter. And also, I'm going to plug the Discord channel. We do have a Discord channel. If you guys would like to join that. And then also, if you're enjoying what you're watching and would like to know when I go live with my experiments, including continuing our cloning procedure that we're going through today, hit the follow button and then the bell for notifications, and it should tell you when I go live next. So if you're interested in more experiments, I'm obviously going to be doing more experiments than this. I'm not just doing this in the lab. There are plenty of other projects that I'm involved in that I would like to stream and show you guys as well. So you can look forward to that, including sequencing. We're going to be doing some sequencing, if you're familiar with that, hopefully. You've heard enough about like 23andMe, where they do DNA sequencing. We're going to be doing that as well in the lab. And I will be showing you how we do that, which is basically a lot of colorless liquids moving into other colorless liquids in tubes. Our favorite thing to do in the whole wide world. And I'm not saying that sarcastically. So yes, if you'd like to join the Discord, feel free. You can talk science, you can talk games, you can talk general chat. I just ask that it remains family friendly because I'd like to remain family friendly. I want this to be educational. And if we have some younger viewers, I just want to make sure that it's safe for them. But yes, feel free to join that. If you miss a stream, I have my YouTube already set up as well. If you want to catch a previous stream, so I only have one since I just started streaming yesterday. But if you missed yesterday's stream, you want to know exactly what I'm talking about as far as these plasmids that we made, you can go back and see that. And I apologize for the focus on the camera. Hopefully it's better today and you can see better what I was showing up at this level. And the breathing. Hopefully that has also changed today in the microphone, as I forgot to set my gates. But yeah, so feel free to join the YouTube and watch the VODs that I put up. So this will be going up on YouTube after the stream as well. if you missed part of it, the beginning, and you wanted to see from the very beginning, go right ahead. But yeah, everything will be going up on the YouTube channel because I don't want you to miss anything. Also, if I refer back to a VOD or something, then it's there, you know what I'm talking about. These also will remain up on Twitch, I believe for 14 days is when they keep the videos up. So if you wanted to go back even on Twitch to look at past streams, you'll be able to do that for a certain amount of time. Feel free to make clips. I would love it. If you join the Discord, you can put the clips in the Discord. I have a channel for that already, and that would be great. I would love it. And I'm working on trying to get the closed captioning set up, live closed captioning for the stream. I'm going to try to get that going for next week in that case, so I can be a little more accessible for my viewers in that case. But yeah, so I apologize, I don't have them right now. But next week I should be able to have that set up. And I know sometimes they can have some funny things show up. So if you notice a closed captioning fail and would like to clip it and share, feel free. Feel free. But yeah, so you can look forward to that next week. But yeah, if you guys are interested in anything else, let me know. Like I said, I've got some new commands today. If you wanted to have a brief intro on what E. coli is, ! E. coli. I also have a little blurb about transformation, ! Transform. Excuse me, I know how to talk. And then the ones from yesterday are still plasmid, ligation, cloning. So those are still up there, just in case you would like a definition of what I'm talking about outside of what I've already told you. It's there and always there. I also have, in case you're new and have no idea, not that one. Excuse me. I know my own things. There we go. What our mascots are. These are our lovely channel mascots, Bert and Ernie, and Eli E.coli. I put that in there just in case people are interested in what they're looking at on my bench. But these are guys we work with. DNA, double-stranded DNA, and E. coli. And I put the little Eli E. coli's tentacles here because I know some people don't like to see tentacly things, so that's why they're down there. I just want to be accommodating for people that it might trigger them, and I don't want that to happen. I want you to be comfortable in this stream. I want you to be able to ask questions. There are no dumb questions. I know people say that all the time, but I firmly believe that it's better you ask than sit there and just let it eat away at you like, oh, I should have asked that question. I should have asked that question. So please, if you got a question, put it in chat. Even if it's about outside stuff, meaning outside of science, you can ask a question. It's all right. We can talk about other things, but please remember to keep it family friendly in that case, and obviously no harassment and bullying. That will not be tolerated. Because like I said, I really want this to be a nice community, a chill community, and so we can just hang out and enjoy ourselves doing science and at the bench. But yeah, so let's see. So that's my little promotion blurbs, basically going on about the community and what I do. Let's see. But yeah, welcome to new people who are joining. If you want to go back and look at this later, if you really want to see what I did earlier, feel free to look at the VOD when it's up on the YouTube or on Twitch when it shows up later. Just in case you wanted to see the beginning of this procedure and you're just popping in. But we got about less than 50 minutes to hang out together. And you get to see my hands. I talk with my hands. And I hope that's okay. But yeah, so any sciencey stuff you guys would like to talk about? Or, like I said, I will geek out about games that I've been playing. If anybody has heard of them or is interested. Because I'm extremely excited for 2 Point Campus that's going to be coming out August 9th. So excited. I love 2 Point Hospital. It's a So 2 Point is part of Sega, and they make hilarious game. 2 Point Hospital is basically you're managing a hospital with crazy patients, crazy diseases. You make rooms to help cure these patients, and some of the diseases are hilarious. Oh, thanks for the follow, Streamer's Life Matter. I appreciate it. Thank you. Oop. I'll have to make the stream label a little bigger since your name is a little bit larger than most of them. But thank you for the follow. Thanks for hopping in. I hope you're having a great day so far, and hope you have a good weekend as well, if it's already the weekend for you. But yes, so I'm so excited to play 2 Point Campus. I've seen a bunch of people with early access play it, and it just makes me even more. And in 2 Point Campus, same kind of comedy showing up, except you are a school administrator and you're building schools, basically college, university. And there's clowns, there's cooking and science, and I've seen robots that are available, magicians. It looks so much fun, and I am just pins and needles waiting to play this game, because I just enjoyed 2 Point Hospital so much. But yeah, so I don't know if anybody else is excited about that or has heard about it before. I highly recommend 2 Point Hospital if you like simulation games and don't mind doing hospital administrative stuff. It's always good for a laugh. And the announcers, because they have a radio in the game. So funny. It is sometimes can be British humor, but it's all right. It's not heavily British humor, just because the studio is over in England. But oh, it's great. And even the radio from the streams that I've seen or the YouTubes that I've seen for Campus sounds amazing. I can't wait to hear all of it and be able to play it, get my hands in there. It's so great. So that's the big game I'm looking forward to. At the beginning of August. Yay! But other games that I play. So last night I actually did do a little bit of gaming. I played House Flipper. So it's one of those chill games. And if you've heard of it, if you've played it, you know what I'm talking about. You basically go through, clean houses, paint the walls, buy furniture, buy plants. Depends on, they have some DLCs that are out for it, where you can do pets now. That was their latest one, so you can get a pet and take care of the pets, and they'll go with you to your jobs. You can demolish walls, which is always fun, demolishing things. But yeah, that's another chill game. I usually like to play chill games. Fall Guys, I will play every once in a while. I'm just really bad at it. It's fun though. It's fun playing with people. I'm just terrible at Fall Guys. It is not the best, but it is always good for a laugh and playing with friends on there too. So that's always fun. But I haven't played it a lot, and I only really played it a couple times because it just became free on the Switch and Xbox PC. So now you can play it for free. And what's great is it's cross-platform, so you can play with your friends on Xbox, someone's on a PlayStation, you can all play together, which I love. I love that. Because most of my friends don't have a Switch. We have a Switch. So it's hard to play sometimes without cross-platform. But yeah, so I play that every once in a while. I'm just terrible at it. So it's funny. It's funny to watch me as I fall constantly. But yeah, the other games, like I said, I like to play simulation games. Maybe that's why I like being a lab manager. Because I like managing things outside of the lab and inside of the lab. It's just a lot of fun to do the management side. Maybe I just like to micromanage, but I promise I don't micromanage in the lab. No, I don't do that. I like it to be nice in the lab. I like to be more of a mentor for the students that are here, so we'll have grad students and undergrad students that'll come through, sometimes high school students, and I like to have a mentoring style with them. So that's one thing I guess I can talk about what I actually do as a lab manager, in case people are interested. Um, so I've been in this lab, this particular lab, for gonna be 8 years. Crazy, crazy, 8 years. And that was right after my postdoc. I got this job, and obviously postdocs after you get your PhD. So I came here, started as a lab manager, and the lab was brand new. So I've been here ever since the lab was started, which is great. And since then, we've had plenty of grad students come through. We've had undergrads. We have a current undergrad. Have we had a high school? I think we've had a high school student in the other lab. I don't think we've had a high school student in wet lab, but that's okay. That's okay. But yeah, so basically my day today is I will do experiments here at the bench. So this is one of the experiments I'm doing. And then I take care of purchasing. So I'm involved in purchasing like this stuff, like our tips, our plasticware. I'm involved in purchasing any equipment that we might need, or office supplies, that kind of stuff. So I do a lot of that. So it's financial. And then mentoring students, so leadership as well. I help them come in, help them start up their experiments, help them start up their projects, help them whenever they need help. If I have the time. Sometimes I have a lot of projects myself, but I try to help them as well, help them learn how to navigate the PhD process, because that can be daunting in itself. So I'm always open for talking about that with the students. Or if they have any, we do a lot of troubleshooting together when it comes to like, oh no, my experiment failed. So we'll talk about reasons behind that, the next steps. So problem solving, critical thinking. I know I'm throwing out like these skills, but that's what you use. Time management is also a big skill to use in the lab. So like I said, we're waiting here for 41 minutes now. Usually if not streaming and talking to you guys, I will be doing something else, like orders, or I might even have another experiment going at the same time. Multitasking. No, I mean, if you can handle it, you can do the multitasking. Sometimes it's not a good thing. I've had experiments go bad because I was very bad at my multitasking during the day. So make sure that you have an idea of what your limits are when it comes to doing procedures and protocols in the lab. So if you know that you can't multitask while you're doing one procedure, don't do it. Don't try it. Don't try it. But with this one, I'm just letting something sit there for a while. So I've got time. So I could be off making the plates that I'll show you once this is done. Or I could be making a buffer that we might need. We're always using, so we use 70% ethanol as a way to clean up the benches when we're done, or clean up any of the equipment that can handle it. Some equipment you can't use 70% ethanol on, so you have to make sure you know where you can use it. Or yeah, basically helping out with troubleshooting, doing work on the computer. So when it comes to the plasmids, and sometimes I will do plasmid design. So I actually use a program to make the plasmids that we are using today, to figure out, one, what will it look like when it's run on a gel, so I have an idea of what I'm looking for, and if something funky comes up, I know what I can look at it, and that is extremely helpful. So that's like the very, very first step, but I can't show you that because that's a specific program, and unfortunately I won't be able to show that. But that's one of the big things that you can do, is work on the computers and basically design your plasmid. So genetically engineer your plasmid that you're going for. Or organizing. Organizing is also a big part of my job. I try to keep my bench organized, but I also organize where we put our back stock of items and paperwork. And sometimes I actually work with sales reps too while we're here. So companies that we buy these from, the sales reps will come in, and I always try to save money for our lab. So I'm always like, do you have any promotions that I might be able to use, especially when it comes to our plasticware, so the tips that we use, because we use a lot of these. We go through a lot of these during our projects, because That's what we use. These pipettes all the time. All the time. I'm always trying to find a deal. So it's always good to know your sales reps. Just in case there's a nice discount coming through, you're like, yes, perfect. That's what I've been waiting for. Or sometimes they can help you, like if you want to buy something in bulk, like tips or the microcentrifuge tubes. So these guys I'm talking about, they might be able to give us a bulk discount too. So know your sales reps. If any of you guys were thinking about being involved in this sort of thing after, if you're doing a PhD or something you are never even heard of being a lab manager before, you can just give me some of the ins and outs of what I do. Let's see, what else? I help on papers, so I will help edit papers that go through. So our lab obviously publishes our research in scientific journals that are out there, so I will help edit the papers. I also help writing grants. So our money and what I get paid on are grants from, right now, government and also internal university grants. So I don't get paid by the university, I get paid through the grants. So need to do experiments, get data, show it to these guys, and they will give us money. So we can keep doing it, so I can keep having my job. So it's a lot, but I enjoy it, I really do. I like being at the bench, I like talking about science, and I enjoy the troubleshooting that goes through sometimes. I mean, sometimes it can get frustrating if it's over and over again and nothing seems to be working. That can be frustrating, and that's when perseverance really push yourself through in that case. And editing, I actually like to edit papers. I do not like to write papers. I will confess this right now. I do not like to write papers. I don't mind editing them, I just don't like the writing process. I've never really liked it. Even in my PhD, I did not enjoy it. But I will do it. I will do it. I will not say no if the opportunity shows up and I'm like, could you please write this? I will do it. I just won't enjoy it. Not as much as editing papers. I haven't really done much figure stuff. I'm usually just at the bench and generating the data for people that will make the figures. So the grad students, or I sympathize with that. Yes, Dizzy. It's a thing that you have to do. You have to write, you have to publish, so you can, as a grad student, get your thesis out, or as a PI, you need those grants. You gotta do it, unfortunately. You have to do it. But if you don't enjoy it, it's not fun. You need that motivation. Sometimes getting that motivation is the hardest part. It's like, I know I'm not gonna like it, but I gotta do it. I gotta do it. I gotta work through this. But yeah, just confessing at that point. Other people really enjoy writing papers, and that's fine. Awesome. Good for you. Good for you. Keep doing it. Keep doing it. And I would love for you to write my papers. Go, I'll give you the data, you can write the paper, and I'll edit it when you're done. That's what I like to do. That's what I like to do. Oh goodness, I lost my train of thought before we got onto the papers. Talking about what I do as a lab manager. Oh yeah, so like I said, I haven't really made any figures myself because I'm usually just doing this. And usually that's grad students or PI, my boss, principal investigator, head of the lab. We'll use it, postdocs, the ones that usually become first authors on the paper. I'm not in it for first author. That's usually when people go for, like, you want a first author paper because that looks good. That means that you did the majority of the work, you did the majority of the writing. I'm just there to help. So I'm not in it for first author papers. In this case, I'm here just to help and get that data going through. And then of course conferences are a big part of science, and that's, I mean, depends on where you are. There's always a conference going on. I know the pandemic kind of slowed that down. They're starting to come back though, which is good because it's nice to be able to go to a conference, show off your work to other scientists who are interested. And by scientists, I mean PIs, postdocs, grad students, undergrads, and sometimes high school students will go to conferences as well. Show off what you got there, like either through a poster or through an oral presentation. Get your name out there. I mean, I know students have gotten postdocs just by going to a conference and networking. as well. Networking. Oh goodness, networking. Talk to people. Talk to people at the conference. You never know when your job is going to come up, your next postdoc opportunity or a job opportunity. Network at those conferences. Meet those people. Meet your colleagues. Do it. Talk to people. Sometimes it's hard. Sometimes it's hard. I understand. I understand. I was a very shy grad student when I first started, but when you find other people that like to talk about what you do and or share the same topics that you're interested in science, it becomes a lot easier. It becomes a lot easier. Or even outside of science, people that enjoy the same hobbies as you do. You can make lots of friends at conferences if you so want to. Would like some new friends. So yeah, I'm excited about the conferences coming back, and hopefully I will be able to go to a conference soon. Not this year, next year. I would like to go to a conference. I haven't been to one in so long, and I do enjoy them. I do enjoy talking about science and other things with other like-minded people. But I also enjoy this. I'm so glad that I get to talk to a wider audience as well. Like I said yesterday, I have been waiting to do this for a long time. Finally got the opportunity to do so. It was like a beacon went off. Because I'm gonna plug it. Streamlabs was hosting a giveaway when I was part of the professional boot camp by the Gamehers, and I won their contest, which awesome. I'm so happy because I got this web camera and a microphone and a subscription to Premium Streamlabs for a year. So it basically was just, it fell in my lap, this golden opportunity. Finally was like, yes, I can do this. I'd been talking about it for so long, but we finally can do it. So now I'm starting. So happy to be here. And for new people that have joined, please, if you want to ask any questions about science, about I was talking about games earlier, about what I'm excited about 2 Point Campus coming out, please put them in the chat and I will try to answer them. If I don't have the answer, I will look it up and get back to you about it. And then, let's see, how many people we got. Oh, we got a good number of people in chat. Hello, welcome. If you would like to join the Discord, you want to keep going, please join the Discord. That'd be great. If you missed earlier on the stream of what I was doing as far as the protocol for the transformation, the VOD will be up on YouTube later. And I'll plug my YouTube again, since those of you who join aren't going to be able to see it. And also up on Twitch as well later in the day. And me talking about the science. So if you have questions about today, you just popped in the stream, you have no idea what I'm talking about from the title or who these guys are, pop in the chat, ask a question, go for it. That's what I'm here for. And I want to talk my science with you guys in this case. But yeah, so that's basically what I do as a lab manager. I mean, there's other things that obviously pop up. I mean, we have a large freezer called a -80. It's basically a freezer that goes down to minus 80 degrees Celsius. So very, very cold. Very cold. Which houses very important samples for our lab. And sometimes, all of a sudden, it will not keep the temperature, and it's a mad scramble to figure out what is going on and moving the sample. So sometimes my day can be very stressful and not exciting in that case. That is not exciting. That is stressful. But wasn't planning on it when I came into work for the day. So sometimes you get little surprises when you wanted to just do your experiments when you come in. So it keeps you on your toes sometimes. So that has happened. That has happened before. Not fun. So you also keep up on equipment in the lab as well. But yeah, so basically that's what a lab manager does. And different labs have different roles. I know that sometimes labs can't afford a separate lab manager. I know staff can be expensive. So postdocs will sometimes take over that, or I've even seen grad students take over a lab manager, like a senior grad student take over a lab management position as well, just to keep the lab going. Because ordering, writing, mentoring, it takes up time. It really does. It takes up time, and sometimes it's time at the bench that you're not going to be able to gotta postpone things. but it's something that has to be done, and I'm grateful that I can do it. I'm grateful that I can do it, and I enjoy it. I definitely enjoy it. But yes, I understand sometimes we can be very expensive as staff. Very expensive as staff. But let's see, what else? Let's see. Any other lab manager things? I'm trying to think. No, that's basically it. That's basically it. But yeah, so, is there anything you guys would like to talk about? I'm gonna put it back in chat, if not. Also, thank you, lurkers. I'm gonna say it again. I always love a lurker. I'm a lurker on Twitch. Just because it's nice to just be in the vibe and watch people do what they do or listen to them talk. I'm all for it. So if you just want to lurk, it's all good. You just don't mind me talking, in this case. But yeah, so let's see. What's something else? Something else we can talk about. I already talked about my 2-point campus, diverging from science again, and how excited I am about that. I guess another game that I'm excited about, and it's simulation games. It's simulation games. Sorry, that's just what I like to play. Is Disney's coming out with Dreamlight Valley that looks like fun. It's going to be a life simulation, and I'm excited about that too. I'm a big Disney fan. I like Disney movies, and with having 2 young kids, we watch Disney a lot. We definitely watch Disney a lot. A lot of movies, a lot of the TV shows, so I've been keeping up with Disney. But you can be your own character in the Disney world and meet all the Disney characters from the movies. So there was someone that was playing, one of the people I watch on YouTube. She was playing like she was able to show 10 minutes of this game. So she met Merlin from Sword in the Stone. And you go around, you do crafting. Looks like there's crafting, there's farming. You can build your own house, you can furnish it. There's character customization at the very beginning. Oh, very excited. Very excited for that one too. I'm more excited for 2-point campus, but I'm excited about having a new life simulation game as well, outside of The Sims. Because there doesn't seem to be enough of those. Doesn't seem to be enough of those. The Sims is fun. It definitely is. But I want some more. I want some more. But yeah, so back to gaming. I'm sorry. It's just one of the things that I like to do outside of the lab. I played House Flipper last night. Just chill. So chill. And then another game that I like to play a lot of indie games, is Let's Build the Zoo. And I've been contemplating, like, eventually I might have part of a gaming where I play this game, because there's science involved in that game. And by science, I mean there's CRISPR. And I know CRISPR has been a hot topic, and like one of those buzzwords that's been going through the science community and outside of the science community for a couple years. And I just want to be able to like go through it and explain it as I play, because you can CRISPR animals to make these weird hybrids out of different animals, and it's fun. It's so much fun. It's a simulation game. It's a management game where you're building a zoo. So yeah. But it's fun. It's fun, and it's very reminiscent of, I might date myself here, Zoo Tycoon back in the day. Very reminiscent of that, and I enjoyed that when I was younger, playing that. So that's another good one. Another indie game. And I'm big on Steam, so all these games are available on Steam, or will be available. I mean, 2-point Campus and Dreamlight Valley aren't coming out until August and September. So yeah, that's another fun game that I enjoy. But I also have so many games on Steam that I haven't played. My library's full of games that I haven't even tried yet. One of the other games that has science in it that's interesting is Jurassic World Evolution. So I'm sure people are familiar with Jurassic Park and Jurassic World. I mean, if you're not, you should be. Get into it. Dinosaurs. If you like dinosaurs, you gotta watch the movies. If you're old enough. But yeah, Jurassic World Evolution also involves science. You're basically engineering dinosaurs in the game. And I'd love to talk about that as I'm playing the game as well. And like, what? Because the original Jurassic Park, guys, you can't just put frog DNA into dinosaur DNA to make up all those holes in the genetic sequence. It just doesn't work. It doesn't work. But it's so fun. It's so fun. with dino DNA. You gotta love it. That was the original Jurassic Park. But yeah, that would be another game. That's also a simulation. Also a simulation. Oh, with dinosaurs. With dinosaurs. Um, and that's a lot of fun. And the science is so good. So good, as in it would be great to discuss as I'm playing it. As I'm playing it. But yeah, we're getting closer. We're getting closer to when I'll be able to plate these guys. So these E. coli have been shaking at 37 degrees Celsius for math. About 40 minutes. About 40 minutes, so a total of an hour. When this is up, we will plate them on the plates that I have warming up to 37 degrees Celsius in our incubator. And the plates have the antibiotic resistance that I was talking about before. So if Eli E. coli takes up the plasmid that we made yesterday, and it should be able to grow on these plates because it has the antibiotic resistance gene in the plasmid. If it doesn't take up the plasmid, it won't be able to grow. So there'll be no Eli E. coli and no DNA. So it'll just be a blank where those cells are put on the plate. So I'm gonna put these guys back. So yeah, so new people. These are our mascots. Bert and Ernie are double-stranded DNA, and Eli E. coli. What we're working with today. But yeah, so if you don't know what I'm talking about, once again for new people in chat, I do have some commands for plasmid, cloning, ligation, transformation, E. coli, just to give you some brief definitions of these science terms. If you have questions, put them in the chat. Not a problem. But let's see. Once again, I've put up the Discord if you guys want to go in there and you want to talk off stream about science, or you have any questions with this and you want to see the other streams, go right ahead. So I'm going to plan on streaming on Monday for a little bit to show you what the plates look like after we do this, and if they're colonies, hopefully there should be. I will show you what we do with those colonies. We basically pick them and then grow them up so that we can get DNA from them the next day. So hopefully I'll be able to also stream on Tuesday next week. So Monday and Tuesday. So I apologize if the schedule is going to be changing. It's just based on experiments, what I'm doing as far as experiments. And this is just the way that it goes. So it won't be every Thursday, every Friday. It'll be different every week. If you want to keep up with that, I will update the schedule on Twitch for everyone, so that if you want to pop into my channel, you're like, oh, when's the next time she's going to be live in the week? I will make sure that it's updated with what we're going to be doing in that case. Also, if you want to know when I'm going live next, hit that follow button and put in notifications. You want to keep up with the procedure that we're going through in the future, go right ahead. This is just one experiment that I'm doing. I'll be doing plenty of others. We're going to be doing some sequencing at one point as well, and a lot of it. So there might be a lot of streams where I'm just doing sequencing prep, and I hope that's okay. But I mean, hopefully I'll be getting new followers, and new people can join us every day, and it'll be new for them at every time. But it'll be a lot of sequencing, I'm not gonna lie. It's gonna be a lot of sequencing, just because that's my next big project that's gonna be coming through. But this will be fun. And if we get the ones that I want, if these plasmids are what they're supposed to be, I'll be even happier, because I'll have what I need for future experiments and projects. That'll be great, instead of troubleshooting all the time. But hey, even if we have to troubleshoot it, you can do it together, and I can go through that process with you in that. And oh, actually, this reminds me, I'm gonna get a setup so when we plate, they'll be all set. So yeah, so any questions? Anything else you guys want to chat about? Or I'm just gonna keep talking to you, if you don't mind it. If you don't mind my talking. Because I don't mind talking either. It keeps you here. Might be good ASMR. I don't know. If my voice is soothing, that's great. That's awesome. I've never gotten that compliment before, but I've also never streamed before in this case. But yeah, I'll just keep talking to you. We got 19 minutes until the procedure's done. So this is probably one of the longest procedures where we're just waiting for something. The sequencing one, we don't have too many long incubations, and they're broken up. It's long throughout the day, like the chunk of it for the sequencing setup, it's like a couple hours, but there's less long incubations involved. And this one is, like I said, usually I'll be doing something else during this time, but today I get to talk with you and chat, and all those interested in the science that I do at the bench. But yeah, so let's see. You can go back to gaming if you want, and I can talk about more games. I'd like to do that. Or TV shows that anybody's interested in. Watch a lot of TV. Watch a lot of TV. Sometimes not just kid shows, but a lot of kid shows, unfortunately, just because of the 2 young kids that I have. Movies that are coming out. I'm looking forward to seeing, maybe eventually seeing, I don't know, might not be able to see it before it gets out of theaters, the Thor movie, Love and Thunder. That looks good. I'm a big Marvel fan. Really enjoyed Thor Ragnarok. I thought that was a great movie. Great movie. And I love myself a good 80s throwback with the way they did it. I just thought it was great. And I know some people have strong opinions when it comes to the Marvel movies, but I thoroughly enjoy them. I haven't had a chance to watch the new Doctor Strange one, but we're getting there. We're getting there. We'll see it eventually. We'll see it eventually. But yeah, hopefully I might be able to see Thor Love and Thunder in theaters before it's out. So that's on my docket of movies. As far as TV shows go, outside of kid shows, outside of kid shows, trying to watch the new Stranger Things series. But that's one of those. So at the end of the day, I confess I'm pretty done at the end of the day after getting the kids in bed and dinner and dishes and laundry. All the chores are done. I kind of just want to chill on the couch, and Stranger Things is one of those ones where I can't just chill. I'm sure if you guys have watched it, you understand. So we haven't gotten past, I think, the 2nd episode. If we've even gotten past the 2nd episode, we meaning my husband and I at home. So, no spoilers. Haven't gotten to the end yet. But we need to watch the end of that. And then, and I know it's scary. I know I've seen briefly that people said that it's starting to get scary. But we're trying to get through that. And it was 2 episodes. Thank you, Tree Wizard. Tree Wizard is my husband. He is graciously joining in the chat, and I appreciate it. Hello, husband. 2 episodes, yeah. So we've got a while ago. We'll eventually do it. We'll eventually get through it, Tree Wizard. I just when I'm there, when I'm able to actually appreciate the show and maybe not have nightmares from it. I know people are saying it's very, very scary. I'm not really big into horror because it tends to give me nightmares when I watch horror movies or something like that. I have enjoyed Stranger Things. I have enjoyed it because, one, 80s throwback, always good to see. Good music too, gotta enjoy the music. But it has been an interesting story, in my opinion. I've enjoyed it, and Tree Wizard and I have had some great discussions about past seasons as well, on how it all works, how like the Upside Down works and everything like that. So we'll get there. We'll get there eventually. What else? Just regular TV. I'm a big Bob's Burgers fan. Love Bob's Burgers. We just watched the movie. It came out on Hulu and HBO Max. So we finally were able to watch the movie that came out. If you haven't seen it, Bob's Burgers fan, wow, great. Loved it. Amazing. Sometimes I will just put Bob's Burgers on in the background if I'm doing something. It's a very good background show since I've watched the seasons multiple times now. It's funny, great jokes, love it. I've even tried to make some of the burgers from the show, because they have a book where you can make the burgers from the show, and I've made I've tried to make a couple, like 2 of the burgers, and some of them you need some specialized ingredients that are not available here where I am. I bet it would be on Amazon. Amazon has everything, right? So I'll have to try those eventually. But yeah, that's one of my favorite shows to just put on in the background when I'm doing something, and it's so funny, so funny. I greatly enjoy it. I greatly enjoy that. So we just saw the movie on that one. That's the latest movie that I've seen. Look at that. I mean, because we have a tiny toddler that hasn't been able to get vaccinated, Tree Wizard and I haven't been able to get out very much. So hopefully that'll change. He's going to be getting his vaccination, and then we'll be able to, do more stuff again. And babysitter. Babysitters are always good too. But yeah, I know I'm behind the times right now. What else? Oh yeah, Game of Thrones. Game of Thrones was also one big, and I know the Prelude is going to be coming out. House of Dragons, if I remember correctly. That is the name of it. So that's coming out on HBO Max. I understand if you can't afford it. Understandable. Streaming services are getting very expensive. But that's coming out end of August, I believe. If I'm right, end of August. Looking forward to it because Matt Smith from Doctor Who is gonna be in it, and I've only seen that actor as Doctor Who. So I'm very curious to see him as a Targaryen. I did enjoy him as the Doctor. So I'm curious to see him in this different role. This is very different than playing the Doctor. So that I'm looking forward to that. I'm actually looking forward to that, and it's definitely one of those ones where I think I'm gonna make myself watch it and like stay awake to enjoy it. I'm definitely gonna do that. So that is awesome. I'm looking forward to that one. Hopefully it's good. But yeah, so we're getting closer. Only 11 minutes left, and then we'll be able to plate our bacteria, our transformed bacteria. So yeah. What else? Is there something you guys would like to chat about? Anything coming to mind that you'd like to chat about? Feel free. Enjoy the mascots, of course. Enjoy the music. Hopefully you're enjoying my voice. Hopefully you're enjoying my voice and enjoying the conversation that I'm having with you guys. I know it's not a lot of chat, but I don't mind talking to everybody. I know Eli E.coli and these guys haven't been around. I can always move around if you want. I can dance. If you're entertained by the moving of the mascots in my hand. But yeah. So that's just a catch up on basically outside of Kids TV, what we've been watching. Oh, thanks for the follow. Let's see. Potato31, I appreciate the follow. Thank you for coming in. Thank you for enjoying the bench and our mascots, Eli E.coli, and our double-stranded DNA. Why do these plushies look so cute? They are pretty cute, aren't they? They are pretty cute. So these are giant microbes, if you're not familiar. Why would a deadly disease be cute? I don't know. It's funny. So these are the ones that I have, but giant microbes have other ones too. So they have like the herpes virus, they have MRSA, and they all look cute. Even for a deadly disease, they look cute. I guess it's one of those things where you can always buy them and give them to a friend and be like, here you go, I got you MRSA. Isn't it adorable? Isn't it adorable? These ones are deadly, but we do have a couple of the dangerous ones. Yes, we do. So at home, Tree Wizard and I have herpes. The giant microbe, a neuron, a white blood cell, MRSA. I'm pretty sure we have MRSA. It's a cape. It has a cape on it because it's a superbug, right? Do we have another one? I'm trying to think if we have another one. I don't remember. I can't remember off the top of my head. So those are the ones we have. I just got these because this is more of what we do. This kind of reminds me of a giant flashback creepypasta. Oh no! Oh no! I'm sorry. I'm sorry. Here. There we go. Oh, there we go. If it's the tentacles, is it the tentacles? Is it the tentacles coming off of the E. coli? I mean, they're not real tentacles. E. coli doesn't have actual tentacles in real life. They're flagella. But I'm trying to keep it so, like, people who don't like seeing that don't need to see it. Don't need to see it. But yeah, so, which also, I know E. coli can be deadly, and those are the wild, that's the nasty one. That's usually 0157. That's the one that you usually see when people have an outbreak of E. coli. This guy, so Eli E.coli right here, this is our lab strain. We're pretending he's our lab strain E. coli. So he's not deadly. Not deadly at all. This is, yeah. And basically, they're designed to just grow under laboratory conditions. And it has flesh, bones, and legs. Oh no! Oh no! Oh, that's crazy. That's creepy. Oh gosh, no. Oh gosh, no. Yeah, that's real creepy. Oh no, into a park for children. Oh, that's terrible. That's terrible. No, no. No, no. Oh, that's horrible. Well, hopefully not. Hopefully not. So I'm sorry if that's triggering. Hopefully it's not. Like I said. Oh gosh. The macroscopic ecosystem living inside it. Oh no. That is nightmare fuel. That is definitely nightmare fuel. Oh my. I have not heard of this before, as Potato. I have not heard of this before. So, yeah, but it sounds creepy and horrible nightmare fuel in this case. But this is nice. This is our nice E. coli. Eli E.coli is our nice guy. Lab form of it. He cannot... I'm personifying it as a he. I should say they. They cannot infect us. They don't have that capacity anymore. They just live nicely in the lab. So usually E. coli is found in your intestine. And they like to live in your intestine. But this guy can't live in our intestines anymore. It's been designed to just be in the lab at this point. So yeah, he's a nice E. coli. He's a nice E. coli. He can't hurt us. He can't hurt us. They can't hurt us. And DNA. DNA is not gonna hurt us. At least Bert and Ernie. The DNA that we're doing today, our plasmids, nope, can't hurt us. We're good. We're good. We're safe. We're safe with these guys. Of a macroscopic version of a generic bacteria. Okay. Okay. I'll have to look this up. I have not heard of it. It's not specified what species. Oh. Yeah, there's a lot of bacteria in the world. A lot of bacteria. I have no idea what the number is of bacteria off the top of my head. I can always look that up. That is an easy Google. But yeah, there are a lot of bacteria. And if it's just generic, I'm sure it just had the generic features of a bacteria in that case, of what people usually see. Oh no. Oh no. Well, thankfully we don't have to worry about that. We don't have to worry about that. I mean, so our E. coli today, the transformation is taking up our plasmid, but that's it. It's only taking up our plasmid DNA. It's not engulfing anything else. Anything else. So we don't have to worry about that. We don't have to worry about that. Oh gosh, let's not. Let's not. That becomes more like a Katamari, if you guys are familiar with Katamari Damacy. Great game. Great game. So much fun. Not a simulation game. One of the games I used to play outside of that, where you just roll up things. It's great. It's a great game. thoroughly enjoyed it. Many hours of fun on Katamari Damacy. Excuse me. Ooh, we're almost there. Less than 5 minutes. Less than 5 minutes and we can play these guys. Play these guys. Awesome. Awesome. So, yeah. Don't have to worry about our E. coli. Our E. coli are nice. Our E. coli are nice. But let's see. Yeah, usually 3 feet? Yeah. Any oh gosh. any organic matter possible. Organic matter. So, inorganic would be fine. Cars and that kind of stuff would be fine. Ah, that's fine. Yeah, that sounds like a horror movie. A horror movie. Definitely. And we don't have to worry about it in real science. If a bacteria got that big, no. It wouldn't be a bacteria at that point anymore. That's a whole new organism showing up. Whole new organism showing up. I mean, bacteria were microscopic, right? I showed you what the tube looks like of the E. coli that are in there. You can't see individual cells like that. Like, this is giant microbes, so this is blown up, obviously, to show the plushie. But yeah, these microscopic guys. Microscopic. You would have to look at, like, plate it. The only reason we can see it on the plate when it becomes a colony is because there's so many of them together forming these little dots. That's the only way you can actually, like, physically see it on a plate. And hopefully I'll be able, and Monday we'll be able to see it on a plate. So that'll be good. Yes, there are a few in real life unicellular macroscopic creatures. Yes. Please don't, I can't think of them off the top of my head. I really can't. It's definitely a Google for me. I'm a geneticist. I usually don't work with unicellular guys outside of bacteria. So I can't remember them off the top of my head. Other fellow scientists, or Espotato if you know what they are. You can say what they are, these macroscopic unicellular creatures that you're thinking of in chat. That's fine. And provide the information to our viewers. Because I honestly can't remember off the top of my head. But it's okay. It's okay. I don't remember. Yep. Don't remember. Google time. We love a good Google. We love a good Google. Yes. So, perfect. Oh, we have 1.5 minutes left. Alright, I'm gonna get my lab coat on. Redonning the PPE. So we can get ready to plate these guys. And then let's put... Oh wait, actually before I put on... We're gonna say goodbye to our mascots. We don't want them to get dirty. No splash. No slash. That would be sad. Alright, let's get the gloves on. Put my lab coat back down. There. Perfect. Alright. And then when this is done, I will go grab our bacteria out of the shaking incubator and the plates out of the regular incubator, and we'll plate them up. And then basically go back in the incubator overnight for them to grow. So I might go out of range in that case, but that's all right. I will be back shortly after I grab those, and I'll show you how I plate them. In that case, in the lab, and how I learned with few resources a long time ago. All right, I'm gonna go grab these guys. I will get back shortly. Alright, I'm back. Oh, here we go. Ah, Protozoa, yes. Thank you for posting that, Potato. Potato31, sorry. Not sure if you want to be known as. Yeah. Okay. It's an amoeba, giant spherical amoeba that ranges in size from 4.7 to 38 millimeters. Oh, wow. Yeah, that is pretty big. You can see it in 1.5 inches. Oh, yes. Filaments out. Okay. Yeah. Interesting. Interesting. Thanks for sharing that. Thanks for sharing that. I appreciate that. All right. So, I'm just sharing. So, these are the plates that I'm talking about. I know they have a shine to them, but basically what's on them. So, we have, you guys can see it. This is solid media. So, this is called agar. And basically, it's just a mix of proteins that the E. coli need, and everything that basically they want to eat, they want to grow on. And I also included the antibiotic on this too. So the antibiotic we're using is called ampicillin, and there's different antibiotics that people use. But on the vector, so the backbone of our plasmid that we made yesterday, has the resistance to ampicillin. So these are ampicillin plates. And then here are our tubes, our guys that have been shaking with the plasmid. Hopefully they took them up, and hopefully they're happy now. Imagine if E. coli was that size. No thanks. Yeah, no thanks. No thank you. All right. So first things first, label. And I'm labeling them on the plate, the side of the plate that has the media towards the top. Not this way, because I don't know if you can see, but there's condensation right here. And if I label the top, it'll be really hard to see below this when I label it to look at what is growing. And sometimes it'll cover up what I'm trying to see as well. So when I look at it, when I take the plate out, it'll be easier to see the colonies this way, so going through the top versus where the media is, because they'll be growing on the top. So the label can also get in the way of that. So best practice is to label on the bottom where the media is facing up. And then I'll put the date. I'm used to doing it. Today's the 29th, right? Yes. It is a Friday. Alright, the labels, flip them over, and I'm actually going to plate the at least it would be much easier to identify, because you can see it. Very true. Very true. Yes, it would be easier to identify the E. coli when it's that big. But yeah, so I'm going to plate the entire batch, just because, as I was talking about earlier, and I'm sure some of you missed it, the plasmids that I made can be finicky, and I want to have them grow as many colonies as possible on this. So what I do, actually, before I do that, I need to grab a couple of tubes. I'm going to show you my trick for plating bacteria. So I set this to 350. That should grab all of the cells that are in the tube. And I'm going to open plate. And then if you guys can see it, it's a little darker now. I'm going to pipe these guys up slowly. You don't want to mix them. You don't want to mix them. They will not like that. And then I put it directly onto the plate like that. And then I take the little plate and spread it around like this. Now, every lab has a different way of doing this. Different way of plating. I've seen it many times. You can catch botulism from using pink sauce. Oh, really? Is that true? You can catch botulism from using the pink sauce? I am not on TikTok, so I've only seen this mentioned a couple of times somewhere else. I think it was on Twitter about this pink sauce. So I have no idea what is in this pink sauce. But botulism, I'm trying to remember. Because you can get, that's why you don't give infants younger than a year honey, because they can get botulism due to the bacteria that can show up in there. But that's interesting. That's interesting. Because one of the pink sauce ingredients is milk and it's shipped in a hot container. What? Yes, it's the same thing in Botox. Correct, Tree Lizard. causes muscle. Yes, it causes muscle paralysis. Botulism. That's what it does. Yes. Ooh, I'm gonna have to look that up, S-Potato31. I am curious about it. It's milk and shipped in a hot container with botulism. Yeah, that's curdled milk. That just sounds like curdled milk. Gross. Why? Why? No, thank you. Yeah, no. Yeah, exactly. It's gonna spoil while it's being shipped. No, that's gross. There's a reason why you don't drink curdled milk. It gets you sick. Oh, no. Why is this a trend? Why is this on TikTok? Anyway, okay, so we plated these guys. They're all set and ready. They're going to be growing on their plates. I flipped them over just because they're not going to fall off. Like, they're not going to drip onto the top. There's not enough liquid for that to happen. TikTok is stupid. There has been some bad trends going on TikTok, like the Tide Pod challenge. Please no. Please no. That will kill you. Please don't do that. Yeah, so, and also you want to do it this way because, like I said, there's condensation that can occur on this side of the plates, and that can drip onto the plate where the bacteria is growing, so on the media where the bacteria is growing, and can actually inhibit their growth. And I don't want that. I don't want that. So that's why I flipped them over onto the media where it's on that side. Alright, so I'm going to go put these in the incubator, and I will be right back. Alright, I'm back. Okay, the plates are in the incubator, and that is the end of the transformation protocol for today. So we got to go through the entire protocol together. Yay! I'm just taking off my lab coat and my gloves since we're all done. And I'm gonna wash my hands. Good hygiene, like I said before. Especially since I was using a bacteria. I know I was using gloves, but it's a precaution. It's a precaution. Just in case. You never know if there's a microscopic hole in the glove or something like that. Which also, you don't want to be using gloves that have holes in them. That does not protect you. Kind of defeats the case of having personal protective equipment. Yeah, yes, the Tide Pod challenge was a long time ago, yes. The pink sauce is pretty much the 2022 version of Tide Pods. Yeah, in a biological sense, definitely. Biological sense. Tide Pod challenge is more organic or inorganic. Wait, no, detergent. It's detergent. Why am I thinking of that? I'm not a chemistry major. Not a chemistry major. I'm also confessing that right now. Biology, life science is my thing. Chemistry, not so much. But yeah, no, don't eat. Thank you, True Wizard. Organic chemistry. Thank you. This is why it's good to have other scientists in chat can answer that for me. All right, I'm bringing these guys back just for funsies, so we can love. Maybe I'll change it up a little bit. The only problem is Bert and Ernie like to fall over. They're not very stable. But yeah, so okay, all right, good to know. Thanks for the shares, Potato. Please don't do the pink sauce. Please don't drink curdled milk. Please don't drink curdled milk. It will make you sick. It'll definitely make you sick. Don't eat Tide Pods. That'll definitely make you sick. You'll end up in the hospital. Please don't. Please be safe. But yeah, so just to wrap up, basically what we did today was we transformed the plasmids that we made yesterday into our Eli E.coli, so into E. coli. They're going to be growing up on the plates, and if there are colonies, which will look like dots on the plate, that means that they were able to take up our plasmid during the transformation process, and we'll be able to move forward with them. We'll be able to see if they're what we wanted in this case. So that's going to happen hopefully on Monday. I'll be able to show you the plates. Hopefully there'll be something there. Fingers crossed, guys. Fingers crossed. And we can move forward with that, and I will show you what we do at that point. Called Cell Lab, where you create Ooh, an Android game called Cell Lab where you create microorganisms. Oh, that sounds fun. I'm always up for a good science game. Always up for a good science game. I will have to check that out. That sounds like fun. You just create them? Like, build them? Like, do you actually do genetics with them? Or is it just, here's a membrane, here's part of the cell? Okay. Yeah, sure. Like I said, always up for a good science game. I'm curious now. There's genetics, I'm very curious. Let's mess with some DNA in a game that we can't do in a lab. In a lab. There's only so much we can do with DNA in a lab. But games, you can do what you want. Assemble different cell types to make something. Oh, okay. Kind of like Spore. If you remember Spore, if you're familiar with Spore, where you take different parts and make a creature. Is it something like that? Also a fun game, although disappointed when it came out. I thought there would be more evolution involved in that one. I thought it was going to be more like, ooh, here's another date. Sim Life, if anybody ever played that back in the day. Loved that game. Maybe that's what actually got me into becoming a biologist. 580 hours! Oh! So you know exactly what I'm talking about. Ah, you are a master. You are a master at the Spore game. Is it like Spore though? Like putting the pieces together to make a creature in this description? So is it similar? I don't remember how many hours I played Spore. It was so long ago. I don't even think Origin has that information on my game anymore. It is! Oh, it is. Okay. Alright, yeah. So I'm familiar with that. I'm familiar with that process. Okay. Man, they need to have more genetics. They need to have more games where you can mess with the genetics and see how it changes the creature because you messed with the genetics. So, Jurassic World Evolution does do something similar to that. Also, it's free! Oh, yeah, because you said it was on Android. Yeah, great. Ooh, I'm definitely going to check that out. Thank you so much. I'm excited about that. Always nice to make my own organism. But yeah. So, if you want to share your description, go right ahead. So before we close up, I'm just gonna do my shameless plugging. It'd also help if I was in the chat. So here's our Discord server. If you'd like to join the Discord server, I'm gonna warn you, I do have Verify on for that, just because I want it to be a safe space for our community. And you know, there's bad people on the internet. But please feel free to join the Discord. If you have any issues with it, because I've only been through the verification process on separate servers, and hopefully I have the settings correct for mine. You'll have to just bear with me on that. I'm not a very good programmer at all when it comes to this stuff. I'm still new. Just let me know in like my next stream, or I'll probably put comments on for this VOD, so you can comment on that for the YouTube if you want to, and let me know in that case. And then, speaking of, I have a YouTube channel where all of my VODs, all of my streams are going to be going up onto the YouTube channel. This will be up later today, depending on how long it takes to upload it, because this is a much longer stream than yesterday. You can catch it there. And I mean, you can subscribe. I'm not going to ask you to subscribe. I'm new. There's only going to be 2 videos on there. But also, if you want to follow my channel here on Twitch, so you can know when I'm going live for the next time. Like I said, Monday, I'm hopefully going to be going live to show you our plates and move on forward. So Monday and Tuesday is my plan for next week. And probably not, we'll probably have to end it there because I'm actually busy, very busy for the rest of the week. But we can follow up in the week after that. But yeah, if you follow me here on Twitch, hit that notification, the bell, you'll know when I go live. You'll get a nice notification. Let's see. One of my creatures is a swimmer with 24 flagella, 12 on each side. Nice. Spits out. Oh man, Gamitate? I probably butchered that. swimmers and fertilizes a plant thing, producing more of the 24. Ooh, by the way, they are part of the same genome. Oh, very cool. Very cool. I like that. I like that it can be like pathogenic in that it can infect. It sounds like it's infecting that plant. Oh, that's cool. That's fun. All right, thank you so much. I'm definitely going to check out this game. It sounds like fun. Like a fun science game. I appreciate that. I appreciate all of you who have joined me today at the bench while I do my science. So, I did my shameless plug. I did my shameless plug for all of you. Appreciate the lurkers. Always. Always. I'm a fellow lurker. And yeah, so I'm probably going to be finishing up the stream here unless there's something else you guys would like to talk about. Because I do have other things I have to do today. As a lab manager, I have many roles that I have to do. I have to do our lab chores for the day, get that ready for the weekend, you know, take care of biohazard, all that kind of stuff. Fun things. But yeah, thank you so much for showing up, for chatting, and I hope to see you next time. Also made a virus that hacks those... You're going through the whole thing. You're going through the whole thing. Oh, that's great. That's great. This seems like actually pretty complex when it comes to the science to it. If you set it up that way. If you set it up that way. Awesome. Awesome. Alright. But yes, I'm going to end the stream probably here. Please join me for my next stream on Monday. You can look at the schedule. Well, not really a virus. Or like a multicellular pathogen. Still a pathogen. Still a pathogen. But yeah, so you can always check my schedule on Twitch. I will update it for you as it goes. And there's a countdown that'll be on there as well. Awesome, thank you. Thank you. So Monday. Might not be at the same time. Probably not the same time. It'll probably be more towards the afternoon, so hopefully you can make it. Maybe we'll get some different viewers at that time. And yeah, this has been great. Thanks for hanging out with me today. I hope you have a good weekend. I hope you have a good rest of the day. And see you next time. Bye.